Welcome to this first of 3 videos on membranes. The purpose of this video is to introduce the fluid mosaic model. We'll be focusing on the structure of the membrane. The next video, the focus is on how that structure, the fluid mosaic model, allows the membrane to actually function, and we'll put it into the context of cells and so on. The information in this one, the fluid mosaic model, depends on an understanding of phospholipids. So if you didn't get a chance to watch the video on lipids where I introduce phospholipids, I'll put a link at the end of this one. It'll be up here, so you can go back and have a look at that video, or you may want to go and have a look at that video before we get into this one. On the board right now I've represented what we can call a cell. There's the cytoplasm here. Extracellular fluid here. Extracellular fluid just means the fluid around the cell. Our cells are bathed in fluid all the time. This blue line is actually the membrane. And the membrane isn't really just a line, it's actually a chemical structure, a combination of chemical structures, and those chemicals are produced as a result of genetics. Cells contain the DNA, which allow it to produce whatever molecules it needs to build membranes. So when a cell divides, obviously there has to be new molecules to become the membrane. So it's a genetic feature. The fundamental biochemical in this membrane is a phospholipid. And if you remember, phospholipids, I represented them before as being a polar end and a non-polar tail, if you like. So this being non-polar and this being polar. And the other thing we have to realize right away is that this is a lot of water in here. Cells are based on water, and there's a lot of water out here. And with having said that, you recognize right away that if you take a phospholipid and you put it into a watery environment, I'll even go this far. Here's a beaker of water, and we throw in some phospholipids. What do you think they're going to do? They're not going to mix, obviously, because this hydrophobic end, the nonpolar is hydrophobic, it's not going to allow it to mix with the water. The hydrophilic end, the polar end, will mix with the water. So what's going to happen, quite simply, I'll exaggerate the size here. If we get the polar ends will actually sit like this, all across the top of the water. Because this nonpolar portion is actually avoiding the water. It's hydrophobic. The polar end, the heads, will mix with water. And if we go a little bit further, we take this, we stir it all up. I'll just draw that again. Here's the water. If we stir it all up so that these things are forced into the water, it's going to look like this. We're going to wind up with the polar ends will be able to mix with the water, but the non-polar ends are going to be clumped together like that. So we have water, remember, is polar, with the polar ends like this. So this is sort of what we're getting closer to what this looks like here. This thing is actually called a micelle. Not a cell. It's called a micelle. So with that information, we can kind of take a closer look at what this membrane might be. Now the problem is, we've got water on the outside and we've got water on the inside. We've got 2 layers of water. So what happens really, I'll just take this away here, is these phospholipids line up so that each side, the outside and the inside, is lined with polar parts of phospholipids. So we get this. We get polar end. I'll just draw a few of them here. You get the idea. One layer of phospholipids, another layer of phospholipids. This could be the width of the membrane. This could be extracellular fluid, which is mostly water, as I've said. This could be the cytoplasm, which again is mostly water. And it works. This works because the polar surfaces of these phospholipids, both layers, are to the outside. It mixes with the water. And this non-polar part in the middle is avoiding the water. And they say that this non-polar actually has an oily nature to it, which makes absolute perfect sense because these are the fatty acid chains here, and fatty acids are oily in their nature. This is called a bilayer. It's a phospholipid bilayer. Bilayer, B-I, bilayer. So membranes are phospholipid bilayers. So this blue line really looks like that. But membranes are more than just that, because this won't allow it to control its function. The fundamental function of a membrane is to control what can go in and out of a cell. So if we go back a step further, this membrane, this blue line, controls what can go in, and what can go out. Well, this doesn't allow it any of those functions. It won't allow it any controlling functions because it's all the same all over, and what's going to get through there? So there's another component of membranes. So membranes are phospholipid bilayers with proteins embedded in them. This description is called the fluid mosaic model. Of membrane structure. Very important because it allows us to explain this type of function of membranes. So let me get rid of this. I'll show you what this fluid mosaic model can actually look like. So let's get rid of this. We'll remember this. I'm going to use this diagram here. Okay. So there's the fundamental part of the membrane, is this phospholipid bilayer, but there's proteins embedded in it. And let's say that this is the cytoplasm on this side, same as we had a second ago. The cytoplasm separates the membrane from the nucleus of a cell, and in the nucleus is all the genetic material, and proteins get made, and some of the proteins that get made may become proteins that actually, I'll use blue because I want to make sure it's distinctly visible here. This could be a protein. That type of protein is called a transmembrane protein or an integral protein. It goes right through the membrane. So let me keep on going here. We could have another set of phospholipids out here. I'll put the name down, transmembrane or integral, in just a second. Another type of protein doesn't have to go all the way through. It could just be on the surface or partway through. So we have this type of feature. So we have surface proteins Remember, the cytoplasm is on this side, so this is the outside surface. There are other proteins that can be on the inside surface as well, but I'll just represent it this way. Or we can have proteins which are what we call integral or transmembrane proteins. Now let's just go back to that picture I erased. Wanted to make sure I had enough room. I'll make it a little bit smaller. This cell membrane allows things to go in and out. Now you can get an idea now that, okay, some things are going to go through features in this membrane, and that's exactly what happens. It's some of the things that cells need. For example, cells have a constant need for oxygen. They have a constant need to get rid of carbon dioxide. They have a constant need to get rid of nitrogenous wastes. They have a need for glucose. Cells always need glucose to metabolize for energy, and so on and so forth. That's how it gets through. Oxygen, carbon dioxide, they're actually small enough they can sneak through the surfaces, or sneak through the structures of these proteins. And we'll get to that in just a minute. So these surface proteins are ones which things can attach to. So it may be that there's a molecule in the environment that has to have an effect on here, but it's too big to get into the cell. And I'm thinking specifically, we'll come up with this one later, things like insulin. Insulin, we've said before, 51 amino acids long. Well, 51 amino acids never going to sneak through here. It's never going to get through. But what it can do, it can bond onto this thing and have an impact on the cell, so that it'll bond onto here and it'll change a feature in the cell. So that's how hormones such as insulin actually affect what's going on in here. Another one that insulin, yeah, okay, I'll just leave that there. I'm going to run out of space here. Oh, I can do it this way. Another example of surface protein would be hemoglobin. If this is a red blood cell, hemoglobin is a surface protein. It's quaternary structure, remember. It's on the outside. It actually attaches to oxygen molecules, and that's how oxygen gets transported through the environment. Things like glucose. Glucose will get through, I've said. But it has to sneak through with the use of a protein. It's too big to just sort of clumsily get through. It has to actually reshape that protein in order to get through. So that gives you a pretty good idea so far of the fluid mosaic model of membrane structure. The next video, like I say, we're going to take a closer look at all of these things and how they work and how things get through the surfaces of the membrane. But before we leave this topic, I have to tell you that there's more to membranes than just this. Because the fluid mosaic model just deals with the protein and the phospholipid bilayer. The fact is, there's more to membranes than just that. And I'll erase a little bit of this so I can illustrate some of those things. I should mention as well that this component, we always say, is the phospholipid part. It provides the fluid nature. The mosaic is the protein part. So if you're looking at how it's constructed, proteins, they are like a mosaic on this, in this sea of phospholipids. And what we'll find out later is that these proteins can actually move about in here. It's not like this one is stuck in that spot. It can actually move, if it's needed over here further, it can move over here, that sort of thing. So that's the phospholipid is a fluid layer, and the proteins form that mosaic within that fluid layer. There's more to it though. You remember the molecule of cholesterol. Cholesterol is a steroid. And as a steroid, it's a big thing. It's like this. It's got that 4-ring structure to it. And, oops, that's right. six-cornered ring, five-cornered ring. There's things attached. That's a fairly big molecule, and there's a part hanging out of it. So cholesterol is an important component of membranes. Cholesterol can actually be found embedded in here. So that could be cholesterol. Cholesterol is a steroid. It's a lipid. It's nonpolar. It'll mix in this nonpolar region in the middle. It's not going to mix too well on the outside. What cholesterol does is it helps maintain the integrity of this thing. Because if you've got things moving around and moving through and that sort of stuff, then there has to be something that helps kind of hold it together a little bit. So it maintains the integrity of the membrane. Our cells themselves don't make the cholesterol. This cell, for example, will not make cholesterol. Our liver cells, if this was liver, it would. Our liver cells make cholesterol, and cholesterol travels through our bodies and gets delivered to cells, and it gets embedded in here. If there's too much cholesterol, what it does, it tightens this up, so it reduces the amount of fluid nature to it. So these things, the phospholipids, can't move as well. If there's too little cholesterol, then they move too much, and the membrane doesn't have that sort of control over what's happening. So this cholesterol is very, very important. Too much, not a good thing. Too little, not a good thing. So we have to be careful about cholesterol levels. Another thing which I can mention about membranes is there's proteins associated, or sorry, there's carbohydrates associated with them. So this might be a carbohydrate chain, an oligosaccharide. Remember, oligosaccharide, oligo, few sugar molecules in a row. This thing might be 5 or 6 sugars in sequence there. It is possible to have an oligosaccharide that's bonded onto the phospholipid. It's also possible to have an oligosaccharide that's bonded onto a protein. So these two things are representing oligosaccharides. If it's bonded onto a protein, it's called a glycoprotein. Glyco is the prefix that's going to imply sugar or carbohydrate anyway, attached to a protein. And if it's on here, it's a glycolipid. So glycoproteins and glycolipids are in membranes as well. These things have the function of allowing cells to have a certain identity. So cells can be recognized by the immune system as saying, okay, that's a cell that belongs here. This other one, which doesn't have those characteristics, doesn't belong here. It's exactly this feature which provides the differences between blood cell types. So if you have blood cell type A, you have a certain combination of glycolipid, glycoprotein things. You have glycolipid, glycoprotein A. If you have blood type B, you have the glycoprotein B. If you had type AB, you would have both of those. If you had type O, you have neither of them. So that's that sort of thing where your cells have a certain identity based on the presence of glycolipids and glycoproteins. So just go back to where I was in the beginning, or not too far from the beginning. I said that membranes are phospholipid bilayers. So they're lipids plus proteins plus more lipids, and now we've got carbohydrates associated with membranes. The only biomolecule that's not a component of a membrane is actually nucleic acids. The others are all there. So I think that sets us up. We understand the structure of the membrane and how that structure is supposed to allow it to function. What we have to look at in the next one is some of the characteristics of the extracellular fluid and the cytoplasmic fluid, and what the needs of the cells are, and how this membrane can accommodate those needs. Like I said, if you need to go back and have a look at the Lipid video is up here. My gecko for subscribing is down here. Thank you very much.